Hello viewers, welcome to Missology. This session is focused on vaccines. Let us first know what is a vaccine. A vaccine is an immunity-inducing preparation containing germ substances. And the practice of introducing a vaccine for developing immunity against a particular disease is known as vaccination. This concept of vaccination was first introduced by Sir Edward Jenner. This was done in the year 1796. Sir Edward Jenner observed that the milkmaids who were in constant contact with the cows that suffered from cowpox never got the attack of smallpox. This led to a hypothesis in his mind, and he wanted to test it. For this, what he did was, he chose a boy of 8 years, took out the fluid from the pustule of a cow suffering from cowpox and introduced it in the body of that boy. As a consequence, the boy developed certain pustules, which later developed into a scab, but he also naturally healed just leaving a scar on his body. But this testing was not enough. He had to test it against smallpox. After 2 months, when the boy was again steady in health, he took the fluid from a patient suffering from smallpox, and now he introduced it in the body of the same boy to see what would happen on his body. This was miraculous. He found the boy never suffered from any symptoms of smallpox and he was healthy and fine. This led to the discovery of the vaccine against smallpox in the year 1796. Similarly, several more methods are known today for the preparation of different vaccines. Now, let's see what are they? The first is where we use kill germs. Now, how can a kill germ ever induce immunity in our body is a big question. But yes, when a germ is killed, it only stops replicating its RNA or DNA, whatever it may contain. But then, when the organism as a whole is introduced in the body of an individual, that recipient's cells will recognize the organism and start developing antibodies against it. This has been used in making the TB vaccine against typhoid and also for producing the SARS vaccine against poliomyelitis. The second method of producing a vaccine is by using the living but weakened germs. Now, how are these germs weakened? These germs are weakened by either the process of heating or by adding formalin to them, which is around 40% formaldehyde that weakens them and makes them almost inactive. So, the recipient who receives it, its body can fight against it well and produce the immunoglobulins or antibodies against it. Now, the BCG, the Bacillus of Calmette-Guerin, which is a vaccine against tuberculosis, is made from living weakened germs. The other diseases where we use the living weakened germs are measles, influenza, where we use the attenuated or weakened germs. We also have cases where we use the fully living virulent germs. And this, when I spoke to you about the discovery of the vaccine by Jenner, it was indeed involved in introducing the fully virulent germs, but definitely in fewer numbers or in less volume. And this is a case where we use it for the production of the antibodies against smallpox. The fourth category is for the production of immunoglobulins using toxoids. Now, what are these toxoids? most bacteria produce a chemical in the body of the individual where it harbors to produce a disease. It's not the bacteria itself, but the poisonous substance that is released by it. And this poisonous substance is inactivated again using some chemicals like formaldehyde or formalin, and that can be used for the treatment of certain diseases like diphtheria and tetanus. There are even other ways of preparing vaccinations where we can use several other methods like the viral vector method. Here in this viral vector method, the vaccination uses a vector, and that vector is definitely harmless. So it is a harmless virus which is used as a vector, and there the chemical is introduced, and here the immunoglobulins that is produced, that is used for the treatment of diseases like Ebola, which is a very serious disease. Another was that when we had the pandemic COVID, during that the AstraZeneca and the Johnson & Johnson prepared the viral vector vaccines against COVID-19 as well. These are few of the very important and mostly widely used types of vaccines. But we have a more recent development in the process of vaccine production where it doesn't use the total organism, rather it uses a subunit or a subpart of the organism for the production of immunoglobulin or antibodies. Here this comes under the subunit vaccines. In the subunit vaccines, the part of the pathogen that we use for the production of immunoglobulins could be very much helpful in certain persons who may not be able to take the total organism in the form of a vaccine. That is, those who are immunocompromised people or older people having some suffering for a longer time, or the very young child whose body doesn't have good amount of blood. So in that case, the subunit vaccines have proved very, very useful. Now, let us find out what are these different categories of the subunit vaccines. Subunit vaccines either use the proteins or the glycoproteins of the pathogens to activate immune response in the recipient. Taking as an example, the hemophilic influenza type B uses a conjugate protein for immune response, whereas hepatitis B uses a recombinant protein type for the immune response to be produced in the recipient's body. The pneumococcus vaccine uses a polysaccharide added with a protein for the production of immune response in the organism. The most recent type is the mRNA vaccine, and this newest kind of mRNA vaccine was developed by Pfizer and the biotech company during the COVID time. Here these vaccines are thought for the most recent development against the production of immunity in a recipient body. So it has a very bright prospect in the future. Thank you for watching. Please do like, share and subscribe to my channel. Thank you once again.